Okay, so now we'll talk about the physical methods of sterilization and disinfection. So the physical methods of sterilization and disinfection are heat, radiation, and filtration. So heat is a simple, cheap, and effective method, and the methods of heat application vary according to the specific use or application. Then you have radiation, which is a very expensive method, so it's only really used in specialized conditions. And then you've got filtration, and although this is relatively inexpensive, it's not routinely used. So as we stated, there are different kinds of heat treatment to kill microbes. First of all, we will talk about pasteurization. And this is the process of heating liquids briefly and then rapidly cooling them before bottling and shipping. And this technique was originally developed by Louis Pasteur, who was trying to find a solution to problems in the wine industry where the wine was spoiling or souring, which made storing and shipping difficult. He discovered that just brief heating of the wine at 55 to 60 degrees Celsius would destroy the microorganisms involved in the spoilage and preserve the wine for longer periods. In pasteurization, the substances are heated in a controlled manner, usually at temperatures below boiling, and this is used for milk, beer, and several other beverages to extend shelf life. It preserves these liquids without destroying the flavour or texture. It is important to note that pasteurization does not sterilize, but it does kill pathogens and drastically slows spoilage by reducing the levels of spoilage microorganisms. There are different methods of pasteurization. So first of all, you have low temperature pasteurization. So that's 61.5 degrees Celsius for 30 minutes. Then you have flash pasteurization. This is also known as high temperature short time processing pasteurization. And this is at 71 degrees Celsius for 15 seconds. And this is what is routinely used for milk. Then you have high temperature pasteurization, which is brief 5 to 10 seconds of exposure to 80 to 85 degrees in continuous operation. And lastly, we have a process where the liquid's heated up to a very high temperature for a very short period. So this is called ultra high temp pasteurization, or UHT, where the liquid's heated to 150 degrees Celsius for 2.5 seconds in a pressurized container using steam injection. And you may have heard of UHT milk. So this is produced by the UHT method and packaged in sterile sealed containers, and these can be stored without refrigeration for months. Although after opening, the spoilage time for UHT products are similar to those of conventionally pasteurized products. Okay, so boiling water can be used for disinfection purposes. If items are placed in boiling water, so 100 degrees Celsius, for 10 minutes, this is sufficient to destroy vegetative cells, so that's the growing and dividing cells, if you remember, as well as eukaryotic spores. But unfortunately, this temperature does not destroy bacterial endospores, which may survive boiling. Therefore, boiling does not sterilize. And disinfection may not be sufficient for certain types of equipment. For example, certain medical equipment will require sterilization, and it will depend on the risk of the procedure they are being used for. For example, surgical instruments used for cutting people open will need to be sterile, so they'll either be sterile single use, where they throw each one away afterwards, or they'll be re-sterilized each time. So next we're going to talk about methods of heat sterilization. So there are two main methods of heat sterilization: dry heat and moist heat. So dry heat is less effective than moist heat and requires higher temperatures and longer exposure times. So for example, Clostridium botulinum spores are killed at 2 hours at 160 degrees compared to 5 minutes at 121 degrees with moist heat. And with dry heat, microbial death is due to the oxidation of cell constituents and protein denaturation. Dry heat is useful for sterilising water-sensitive materials, for example glassware and metal instruments, as it does not corrode them like moist heat does, but it cannot be used for heat-sensitive items like plastic and rubber. So the guidelines for hot air sterilisers are 180 degrees for 30 minutes, or 160 degrees for 120 minutes, whereby the objects to be sterilised must themselves reach these temperatures for the entire prescribed period. Okay, so now we'll talk about moist heat sterilisation. So this is carried out with the use of saturated steam under pressure, and a device called an autoclave is used for this, and it basically acts like a sophisticated pressure cooker. Moist heat sterilisation is effective against all types of microbes. It denatures protein, nucleic acids, and microbial membranes much more easily than a dry environment. To destroy bacterial endospores, the temperature needs to be above 100 degrees Celsius, which is why the autoclave is used. And this slide shows the different temperatures that the autoclave needs to be heated to kill different microorganisms. And as you can see, a much higher temperature is needed to kill the bacterial spores. Okay, so you can see it's 0.5 to 12 minutes at 121 degrees to kill bacterial endospores. And just remembering back to the prions, if you remember, these are even more difficult to kill than the bacterial endospores. So the prions, if you remember, require autoclaving at 121 degrees for 4.5 hours or 134 degrees for 30 minutes. Okay, so prions are even more difficult to inactivate. So this next slide shows the structure of the autoclave. So with the autoclaving, steam is released into the autoclave chamber. The air initially present in the chamber is then forced out until the chamber is filled with saturated steam and the outlets are closed. Hot saturated steam then continues to enter until the chamber reaches the desired temperature and pressure, which is usually 121 degrees Celsius and 15 pounds per square inch of pressure. And if we look next at low temperature, so low temperature inhibits microbial growth by slowing down metabolism. And examples include refrigeration and freezing. So refrigeration slows the growth of microorganisms and keeps food fresh for a few days. Then freezing at -10 degrees stops microbial growth, but generally does not kill microorganisms. And this keeps food fresh for several months. So next we'll move on to radiation as a physical method to disinfect or sterilize. And there are two types of radiation, non-ionizing and ionizing radiation. So non-ionizing radiation includes ultraviolet or UV rays, and the antimicrobial activity of UV light depends on the length of exposure and the wavelength of UV used. So the wavelengths that kill microbes lie in the region of 260 to 270 nanometers, where it's absorbed by nucleic acid. So the UV light is absorbed by microbial DNA and causes adjacent thymine bases on the same DNA strand to covalently bond together to form dimers. This then prevents DNA replication and transcription. However, UV radiation does not penetrate glass, dirt films, water, and other substances effectively. Because of this disadvantage, UV radiation is only used in a few situations. So UV lamps are sometimes placed on lab ceilings or in biological safety cabinets to sterilize the air and any exposed surfaces. So next we'll talk about ionizing radiation. So ionizing radiation, unlike UV rays, penetrates deep into objects. And ionizing radiation ionizes water and other molecules to form free radicals that can disrupt DNA molecules and proteins. And this type of radiation destroys bacterial endospores and all microbial cells. but it's not always effective against viruses. And there are two types used. Gamma radiation, which consists of electromagnetic waves produced by nuclear disintegration, and corpuscular radiation that consists of electrons produced in generators and accelerated to raise their energy level. The downside is that radiocellularization equipment is expensive. It's used on a large scale in the pharmaceutical industry to sterilize things like bandages, suture material, and heat-sensitive pharmaceuticals. It can also be used in the food industry to prevent spoilage in seafood, meat, poultry, and fruits. And the required dose of radiation, ionizing radiation, depends on the level of product contamination, so the bioburden, as we discussed before, and how sensitive the contaminated microbes are to the radiation.